Okay, so we're going to talk about something called allosteric regulation. So allosteric regulation is where binding at somewhere other than the active site affects the protein function. So we have, let's just draw real quick. Let's say here is hemoglobin. Okay, so here are the 4 subunits of hemoglobin. We'll say 1, 2, 3, 4. And there's really 2 alpha and 2 beta subunits. So there's an alpha subunit here, an alpha subunit here, a beta subunit here, and a beta subunit here. So we have essentially 2 dimers of alpha and beta to make up this tetramer, 4 subunits of hemoglobin. So in the center of each of these subunits is where the oxygen binds. So where the oxygen binds, this is the oxygen binding site. We could also call this the active site. It's where the oxygen is binding. The allosteric site is anything other than the active site. Any location other than the active site. So allosteric regulators are something that binds somewhere other than where the oxygen binds, and that's going to affect hemoglobin in some way. So we're going to talk about 3 allosteric regulators. Three allosteric regulators of hemoglobin and what happens. The first allosteric regulator that we're going to talk about is something called 2,3-BPG. And what 2,3-BPG is it's 2,3-bisphosphoglycerate. Bisphosphoglycerate. We're going to see something similar to this when we're talking about the steps of glycolysis in a few weeks when we start talking about metabolism. So keep this in the back of your mind for later because we will come back to this in a few weeks. 2,3-BPG fits in the deoxyhemoglobin pocket. It fits right in here, 2,3-BPG. So this is between the 4 subunits of the hemoglobin, and what happens is, so 2,3-BPG fits in the deoxyhemoglobin pocket, and when it does this, it stabilizes the T state. And remember what the T state is. The T state is the deoxyhemoglobin. So when this 2,3-BPG fits in here, it prevents the hemoglobin from conformationally changing to move that heme in the protoporphyrin ring so it can bind oxygen. So this is allosteric regulation. It's regulation at a site that is not the oxygen binding site. This stabilizes the deoxy state. So there's a lot of research about 2,3-BPG. One thing that is important to mention is that people who smoke, they have a lot of 2,3-BPG in their blood. That means it's harder for hemoglobin to be picked up in the lungs. So not only does smoking impact just the function and the quality of your lungs, but smoking also increases the concentration of 2,3-BPG, which means even when your lungs are working as hard as they can, the hemoglobin is having a harder time picking up oxygen. So 2,3-BPG is an allosteric regulator that stabilizes the T state and causes oxygen to be released because it can't hold the oxygen. So that's the first allosteric regulator that we'll talk about. The second allosteric regulator that we'll talk about is the hydrogen ion. So remember the relationship between hydrogen ions and pH. An increase in hydrogen ion concentration is a decrease in pH. Decrease in pH means more acidic. Because remember, it's the negative log of the hydrogen ion concentration. So the more hydrogen you have, the lower your pH, the more acidic you are. So this happens because when we are going through cellular respiration, we're breathing in oxygen, and the waste product that we're producing is carbon dioxide. Well, carbon dioxide is a waste product and we need to get rid of it. We don't have a carrier protein for carbon dioxide. Carbon dioxide goes through a buffering system called the carbonic acid buffering system. This is all drawn out in your lecture and in your textbook. Where as carbon dioxide is being produced, it's increasing hydrogen ions. So carbon dioxide is increasing the hydrogen ion concentration, which is decreasing the pH. This is also going to stabilize the T state and tell us to to release oxygen. So when you're breathing and you have a lot of carbon dioxide building up in your cells, that tells your body there's too much carbon dioxide here. I need to exhale, release the carbon dioxide, and then I need to release the oxygen in the tissues. So this is homeostasis. This is regulating the amount of carbon dioxide and oxygen that we have in our cells. And this is done by this allosteric regulation of the hydrogen ions binding somewhere other than the active site and causing hemoglobin to release oxygen to the cells that we need it. This is also called the Bohr Effect. And you'll see this in your textbook. This is the same fractional saturation that we looked at before when we looked at the binding curves of hemoglobin versus myoglobin. So what this shows is this would be a higher pH. We'll say pH 7.6 and then 7.4 and 7.2. But essentially what this says is as the blood pH gets more and more acidic, the attraction to oxygen is lower and lower. So if this was the, we'll say 37 where the muscles are, we at a lower pH, we would release this oxygen sooner. This is called the Bohr effect. And this is how hydrogen ions impact the affinity for hemoglobin to oxygen. The last allosteric regulator that we'll talk about is carbon monoxide. So carbon monoxide is 250 times, has a hemoglobin is 250 times more attracted to carbon monoxide than oxygen. So hemoglobin binds oxygen. It also will bind carbon monoxide 250 times more strongly. This is how people can get carbon monoxide poisoning. even when there's oxygen around. So carbon monoxide, sorry, I should have separated these, is not an allosteric regulator because it's binding at the active site. But the reason that carbon monoxide poisoning is so dangerous is because that you can essentially be starved of oxygen and die even when there's plenty of oxygen around, because it takes very little carbon monoxide to get carbon monoxide poisoning. Because carbon monoxide is a really good fit. There is a really strong affinity for carbon monoxide in these 4 active sites of hemoglobin. So even a small amount of carbon monoxide in the environment that you're in could cause carbon monoxide poisoning because oxygen has such a lower affinity than the carbon monoxide does. So the way that you fix carbon monoxide poisoning is you have to increase the partial pressure of oxygen. You're essentially out-competing the hemoglobin binding sites. You're flooding the hemoglobin with oxygen to outcompete that carbon monoxide that's bound there. So see, these are some examples of molecules that impact the binding of hemoglobin and oxygen.